Hello friends, in my series about life in space, this is gonna be the most important video about amino acids, and it's gonna take a couple of turns. The first thing is we have to do some basics, and the basics are not easy, I have to warn you, but for many people learn this at school or at university anyway, but for the ones who haven't, it will be quite a harsh way through, but at the end will be a big reward. So first we're going to look at amino acids and translation, which is a very important process in biology. Then we will shortly talk about handedness or left-handedness, because it's important for our argument, life in space. And then we will talk about amino acids found in space, actually on a very specific body in space. You already saw it shortly on the title slide. And then in the end, the important conclusions, what have we learned for life in space? And I can tell you it's a lot that we can learn from amino acids, but not only from them. So amino acids are parts of peptides or proteins. It's a very just definitional thing. Short chains are called peptides and long chains are called proteins. And proteins are very important for biology. So what are these amino acids that make up the proteins? They are relatively short molecules consisting of an amino group, which is a nitrogen and 2 hydrogen. It looks a bit like a head of an ant or something. And then they have a carboxyl group, which is considered as the end. And carboxyl group means it's like an acid. This part is the acid part. So amino and acid. And in the middle, there's just one carbon atom with a hydrogen and a side chain. And the side chain here is just shortened as R for rest or remain. But this is of course the most important part of the amino acid, because there is the variation and there is the action of the amino acid together with the other amino acids in the protein. And in the bottom here, I show you how 2 amino acids are bonded. So they're just the acid part and the amino part are gonna fuse, and then you have a chain of 2 and one water goes away, a water molecule. Of course that can also be reversed, and the water goes in and the whole thing splits up. But that probably needs enzymes. Proteins are often enzymes. Actually, 2/3 of the proteins or so, or even 80%, are catalyzing chemical reactions. And they're really good at that. Parts are also for structure. So we consist of proteins, and they build us. I won't talk a lot about these side chains. Here you can see a sulfur, and the sulfur is good for bonding with other sulfurs, which gives the whole protein structure then. That's something I shortly wanted to mention, but other rests are doing other things. And here you can already see in almost all of the elements of these 6 magical elements that we consist of. The only one that is lacking here is phosphor. But we have carbon, nitrogen, hydrogen, oxygen, and sulfur. Of course, we consist of more atoms than that, but 99.9% is these 6 atoms. And funnily enough, they are the most often found in space after supernovae. And even stars that don't go supernova, they still make carbon. Not any heavier elements maybe, but carbon, yes. So, and here we see more or less the zoo of amino acids. Actually, there are 20, and you can order them by various criteria. Here they order them if they are polar or charged, and take out some special cases. And here they order them by being neutral, acidic, or basic. So you could probably also take other orders. So these are more or less the letters that build the proteins. With the letters, I will have to talk about the code later. First, a short side issue. For protein, there's a primary structure, a secondary structure, a tertiary structure, and even a quaternary structure. This is not so important for our argument here, but the thing is, whenever you have a protein in solution, it looks like a long thread, like this. And here you can see the structure parts and just the rests, the side kicks that make up the active part of the amino acids. Then the whole thing starts going into characteristic forms, which is either these helical forms or sheets, flat sheets. And most proteins consist of these two, but often they have both. And this is the way how protein makes what it does with this structure. So you can expose certain amino acids in combination and keep them together in a form so they build maybe a tunnel or something that can do something chemical, things like that. And the tertiary structure is rather the three-dimensional structure of the protein in the cell, which you normally can't see. That's a big step from secondary or primary to tertiary structure. There's no proper way of measuring what the tertiary structure looks like. There are some softwares that predict it, but it's not fully clear for each protein. And as one, if you analyze it, you see just that. You can have the chain and extract everything chemically. That's easy. But from this one, you can't tell this tertiary structure. The quaternary structure is for me something artificial, because then you have two proteins and they combine. That's what the quaternary structure is. But often two are building something, a cell, part of the cell, a receptor or something. So, but that was a side kick. Now we have to talk about the genetic code. This one, because you have always three letters, this letter, this letter, and this letter, and they combine to an amino acid. And here are the codes for the amino acids. They are from the alphabet, and because there are 20, the alphabet is enough. but sometimes the letter is the starting letter of the chemical, sometimes not. And the thing is that there is one amino acid that starts a chain, which is the methionine. And there are 3 different ones, or at least 2, that can stop. So the chain will be cut after these. This is part of the code. And the others are the different amino acids. And the ones that the plants and animals consist of are pretty normed. I think there may be some differences for the bacteria, but for the higher life on Earth, they are the same. But one important difference is we can build some of these amino acids, but not all of them. And the ones we have to take up with food are called essential amino acids. By the way, these letters, there are 4 letters, or actually 5, but 2 of them mean the same. There is adenine, there is thymine, cytosine, and guanine. And always 2 fit together. So these 2 and these 2 fit together. These ones I mentioned count for DNA, but in RNA, the thymine is replaced by uracil. But that doesn't have to bother us, because they mean the same thing. They are translated directly from thymine into uracil, actually transcripted, not translated. I'm not talking about DNA a lot, because this is not what we need for the further videos and the further discussion of life in space. The RNA world is much more important. I'm going to show you that in a bit. Now, this thing is not one-to-one. We have 20 amino acids, but we have 64 possible combinations of these 3 letters. And here you see that often 4 combinations build the same amino acid. For example, here even 6. And often the last letter is not so important, because you can have an uracil, a cytosine, an adenine, or a guanine as last letter, and you always get the same amino acid. This is called redundancy. So even if there are some mistakes, don't even change amino acids. And RNA makes mistakes. This happens. We could discuss that later, that mistakes can even be important for adapting life, for the variation, to speak with Darwin. Some codes, the stop has 3 different combinations, so it wasn't completely wrong when I said 3, because yeah, this one, and this one. And actually none has just 1, except the methionine, which is the stop code. Yeah, that's the genetic code, more or less. You can plot it as a circle, or you can plot it as a table. It's the same thing. Now, what do we need that for? This is a cell, and it has a nucleus for the higher animals and plants. So what happens if you need a protein is, first you have to open the DNA. The DNA is not free, it is wrapped around special proteins that can open the DNA up. And then it is transcripted into RNA. And now we have to be cautious, because RNA can mean 2 different things. This one is a messenger RNA. It is just done in the core, and then it goes out into the cell until it finds a ribosome, and then it's going to be translated into a protein or into several proteins. As long as the RNA lives, it can build proteins. But quite soon it will be degraded and killed, and it doesn't go back as a whole. The amino acids are recycled, but the whole chain is broken at some stage. But the thing is, the ribosomes take the chain and translate it into a protein. And they start, and at the beginning, first they look for the beginning, and then they just add amino acids and they bond, and in the end you have the chain. You could wonder if this is an efficient process, because it needs these tRNA. They are holding an amino acid up there. So the amino acids, they are just swimming around in the body, and these tRNA, they are specialized on one specific amino acid and capture it and go to the ribosomes, and if they're needed, they just look for their combination. Actually, they have the reversed combination here on the foot, and then if they find their combination, they bond and hand in their amino acid to the chain, and the chain gets longer. This is called this 3-step or 4-step process: initiation, elongation, termination, and recycling. So the amino acids will be, not the amino acids, the tRNAs will be recycled, because they can go back and look for their amino acid, and once they have it, they will return and look for to be expressed on mRNA. And the ribosome, I always thought it was a protein, but it is not. It's actually a piece of RNA, which is interesting. And this is one of the turns that I promised you in the beginning. Maybe I brought this too early, that's possible, because I'm so much aroused by this this turn here, because it can show us something. So here I'm showing you the thing I tried to explain. The ribosome is looking for the beginning. This is called the 5' end, and then it runs from the 5' to the 3' end and connects these messenger RNAs, and then they bond together in the exact same order as marked here on the RNA. And in the end, the process is terminated and the components are recycled. Of course, not the amino acids, because they're in the protein then. But that's more or less what I told you here. It's just shown better. So then it starts from the beginning, as long as the messenger RNA is intact. This process is actually simpler than what happens in the core here, because the transcription can have more influences and promoters and alternative splicing and all these things, but they don't have to interest us now for the moment. Maybe later, but not in this video. So that's more or less how this translation works. And this mechanism is important because it's basically the same for all living things on Earth. So the this is actually the best relation marker to us. And it can be found everywhere on the Earth, but once we go into space, probably not. If this was the invention that more or less made proteins possible, and proteins are highly efficient for many things. Then the amino acids, most of them have two types, left hand and right hand. You could use a mirror, and then the left hand goes into right hand. This is something that shouldn't happen. But it does happen in certain special cases, but it shouldn't happen. So if you look here, they just put the carbon and the acid rest on a line, and then you can see that one time the amino goes to the right and one time it goes to the left, and the rest goes to the left and here goes to the right. So if you try to put this one on this one, you will realize it's not possible, and this also means that chemically they can't talk to each other. So if you have an enzyme that works on that one, it won't work on that one, and vice versa, because enzymes have to have the right three-dimensional structure, which is the tertiary structure, as we have seen. And the funny thing is that almost all amino acids are left-handed, except one, which is glycine, and this is not handed at all. So it's symmetric. It looks the same if you mirror it. But none is right-handed. By the way, for fatty acids, that's a similar thing, but just the other way around. And probably this also has a reason. But for the proteins, it is also a marker, because if you are looking for biologically built amino acids, they will be on a planet, they will be of one type, either left-handed or right-handed. It's more or less accidental which one. So on the Earth it's just that, but it could be that on another planet, but you won't find 50-50. And this is something you have to mind, because we'll use it later. So, and this later is approaching fast. There are only a couple of bodies where material has been taken and brought back to the Earth. And one of the most important missions is to look for organics on bodies in the solar system. For a long time people thought comets would be a good place, but now they thought also some special type of asteroids, especially the chondrites, could be interesting. The ones that contain lots of water. And now we know that there are some that contain lots of water. And this is Bennu, and Bennu was visited by a probe called Osiris-Rex, which is a funny thing because it reminds of an Egyptian pharaoh. This Osiris-Rex went close to Bennu and then gave a puff, so an air puff on Bennu, and because Bennu is a big chunk of gravel, some gravel went back into the capsule and was closed in, and the capsule went back to the Earth and landed in the Mojave Desert, and scientists made sure that nothing can enter this capsule before it's in the lab. So this is the complicated thing once the capsule is back. And the capsule looks black. That's not accidental, because it got heated by the atmosphere. And the amazing thing is there were amino acids found on the probes from Bennu. a lot of them actually. Most of these 20, histidine, asparagine, arginine, and just counting 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. So some of these are even special ones that do not occur naturally. And also 19 that aren't even coded by genetics. And the important thing is there were left-handed and right-handed, more or less 50-50, which is what you would expect if there is no biological origin of these amino acids. And this, if it turns out to be true, is revolutionary, because you can just put some water and some chemicals on a planetoid, on an asteroid, and let it run through the solar system, and there is some ultraviolet making some chemical reactions, and what turns out is amino acids. They probably didn't come from Mars, they just built here on this asteroid. And the end is also revolutionary. All 5 nuclear bases have been found in the samples of Bennu. So adenosine, thymine, uracil, guanosine, and cytosine. And also GABA, which is found in the brain a lot. If you want to read the paper, that's Clavin et al. Nature Astronomy 2024. So quite a new paper, because it took a bit of time to analyze the samples once they were back on the Earth. That's the revolutionary thing about these probes. This probe. It's really a probe. It's a probe that went into space, and it's a probe that went through the atmosphere. So it's a probe within a probe. Takeaway, that's a lot of takeaway actually. The RNA world figured out how to use amino acids to build proteins. People for a long time were not sure what was first, the proteins or the RNA, until people found that RNA can also build enzymes. And actually, the ribosomes are one of these enzymes. They're not very good enzymes, not very efficient compared to protein enzymes, but they work. So it's possible that RNA was actually first. And if you find these ones on Bennu, you could also have found them on the Earth once it was new and didn't have an oxygenous atmosphere back then. Still quite warm maybe, in the hot ponds or somewhere in the sea. Probably not on the land. Many of them were just around on Bennu. We don't know about other comets or asteroids, but on Bennu they were there, just hanging around. All the amino acids you need, and even the nucleic acids for the RNA. And even for the DNA, although for the DNA you need the strands, and it's a bit more complicated. The protein world turned out to be more efficient, so the RNA world built the protein world and stayed there, and there were more and more protein enzymes and less RNA enzymes. So it was easier for the RNA to give the work to the proteins. I already mentioned ribozymes, and RNA, ribozymes. Enzymes made by RNA are called ribozymes because of the ribosomes. And this is a relatively new thing. I always thought it was a protein, but it's obviously not. The chirality can be used as a biomarker. I already explained that. So everything on the Earth is left-handed when it comes to amino acids. But on Bennu, it was 50-50. So probably it wasn't biology that built these amino acids. These could be seen as a proof that life has a common origin. The life could have been built several times and parts could have been died out, but the line that goes to us is probably one line. The line that is using left-handed proteins and the line that is using this ribozyme to translate because it's needed. We have only one. And it does that. I don't know how fast it does that, but it does that. Build proteins from messenger RNA and transfer RNA. So the mechanism is the transfer of RNA and this ribozyme. And the genetic code is universal when it comes to the Earth. And there are some changes for bacteria, but for eukaryotes, it's the same for all of them. And that we found them on Bennu could mean that the first amino acids could have come from space. And here I just say along with the water, because there was some water on Earth, but never as much. And the Earth was hot. So once it cooled down, it had to get some of the water. And in the solar system, there's a lot of water, but actually more outside than the Earth is. So some of the water came back in to the inner solar system. This is something we may also discuss at some stage. I tell this everybody who comes to the observatory whenever we look at Jupiter or Saturn. And yeah, that's if you compare the planets, always the big riddle, why does the Earth have so much water? Mars still a bit and Venus almost none. Why? Special. So that was this presentation about amino acids. And obviously the first part was a bit technical, but in the end we could get the yield of the technical part. Thanks a lot for the attention.